The paper introduces a novel non-autoregressive generation framework called TabEAE, which extends the prompt-based event argument extraction (EAE) model PAIE to extract arguments of multiple events concurrently. This approach reformulates EAE as a problem of table generation, leveraging the encoding, prompt construction, and span selection modules from PAIE. It also discusses three training-inference schemes: single-single, multi-multi, and multi-single, with multi-single performing best on ACE, RAMS, and WikiEvents benchmarks, and multi-multi on the MLEE benchmark where nested events are prevalent. The research highlights the importance of considering event co-occurrences in EAE, as it improves the model's ability to distinguish the semantic boundary of each event and enhance its performance in extracting single events. The study contributes to the field by bridging the gap between event extraction, EE, and EAE research, and outperforms state-of-the-art EAE methods on 4 benchmarks. It also introduces the concept of text-to-table, a relatively new problem in the area of controllable natural language generation, and utilizes a non-autoregressive table generation framework to solve it. TabEAE consists of 4 components: trigger-aware context encoding, slotted table construction, non-autoregressive table decoding, and span selection. The trigger-aware context encoding component marks each trigger with a pair of markers and tokenizes the marked text into a sequence for encoding. The slotted table construction component initializes the representation of the table by concatenating the encoding of the column header and the initial representations of the argument slots. The non-autoregressive table decoding component updates the representation of the input table via structure-aware self-attention and cross-attention between the table and the encoder output. The span selection component transforms each representation vector into a span selector, which is responsible for selecting a span from the text to fill in the argument slot. This process involves computing logit scores for start and end positions, then calculating the score for each possible span and identifying the optimal span using the argmax function. The research paper introduces TabEAE, a novel approach to event argument extraction (EAE) that leverages a large-scale pre-trained language model (PLM) and a specifically designed decoder to extract event arguments from unstructured text. The authors evaluate TabEAE on 4 benchmark datasets, including ACE05, RAMS, WikiEvents, and MLEE, and compare its performance with several state-of-the-art SOTA methods. Experimental results show that TabEAE is competitive with SOTA models, achieving high scores in terms of strict argument identification F1, ARG-I, and strict argument classification F1, ARGC, on all 4 datasets. Notably, TabEAE outperforms SOTA models on ACE05 and WikiEvents when trained to extract all events in parallel during inference. The authors analyze the effect of the training-inference scheme on the performance of EAE models, finding that TabEAE benefits from extracting all events in parallel during inference, especially on datasets with a high proportion of instances with multiple events. The paper explores the concept of event co-occurrences in EAE and proposes a novel text-to-table framework, TabEAE, which can extract multiple events in parallel. The authors hypothesize that TabEAE can better capture the event semantic boundary when trained to extract all events concurrently. Experimental results verify this hypothesis, showing that TabEAE performs better when trained to extract all events concurrently, with significant performance gains on extracting the arguments of overlapping events. An ablation study is conducted to verify the effectiveness of different components of TabEAE, including the structure-aware attention mask, precomputed encodings of the input table, and handcrafted prompts. The results show that these components are essential for TabEAE's performance. A case study is also conducted to illustrate the effectiveness of TabEAE in capturing the event semantic boundary, demonstrating a stronger capacity to capture the event semantic boundary compared to PAIE. The authors acknowledge the limitations of their work in event argument extraction, emphasizing the need for further exploration in iterative extraction, coarse learning, and automatic prompt construction. They highlight the reliance of their model's high performance on manual prompts, which restricts its application to scenarios with high-quality prompts. The paper references various studies on event extraction, including document-level event argument extraction, multi-sentence argument linking, and deep neural networks for biomedical texts, as well as research on language models like BART and Roberta. The authors stress the importance of understanding entity co-references, a phenomenon often overlooked in narrative documents, which could impact the model's ability to determine underlying relations between entities. They propose addressing this limitation in future studies and highlight key points for improvement, including developing more efficient and effective models, better understanding event and entity relationships, and creating robust evaluation metrics. The paper provides a comprehensive overview of existing datasets and hyperparameter settings for document-level event argument extraction tasks. 4 datasets are profiled: ACE05, RAMS, WikiEvents, and MLEE, with statistics including the number of event types, arguments per event, and events per text. The authors discuss their hyperparameter settings, adopted mostly from Ma et al. 2022, with manual tuning, and present the trial intervals and final configurations in Table 6. They experiment with different numbers of encoder-decoder layers, finding that 17 encoder layers and 7 decoder layers are optimal. The prompt construction process is described for each dataset, with direct adoption from Li et al. 2021, and Ma et al. 2022, for ACE05, RAMS, and WikiEvents, and manual construction for MLEE. The authors provide a thorough analysis of the datasets, including statistics and preprocessing procedures, highlighting the importance of hyperparameter tuning and prompt construction for achieving optimal performance in EAE tasks. The MLEE dataset, as outlined on page 13, presents a comprehensive set of natural language prompts designed to capture various biological events and processes. These include cell proliferation, development, blood vessel development, growth, death, breakdown, remodeling, synthesis, gene expression, transcription, protein processing, DNA methylation, metabolism, catabolism, phosphorylation, dephosphorylation, pathway participation, localization, binding, regulation, positive regulation, negative regulation, and planned processes. Each prompt is structured to describe specific events or entities at particular sites, allowing for precise identification and analysis of these biological phenomena within the dataset. This meticulous construction demonstrates an understanding of the complexities inherent in biological systems and the importance of capturing this complexity in natural language processing tasks. By providing a standardized framework for describing these events, researchers can better analyze and understand the relationships between different biological entities and processes, contributing to advancements in fields such as drug discovery, disease modeling, and personalized medicine. The use of natural language prompts enables researchers to leverage recent advances in artificial intelligence and machine learning to identify patterns and trends within large datasets that may not be immediately apparent through manual analysis. This could lead to novel insights into the underlying mechanisms driving various biological processes and potentially uncover new therapeutic targets for treatment of diseases. In conclusion, the MLEE dataset, along with its corresponding natural language prompts, offers a powerful tool for researchers seeking to explore the intricate relationships between biological entities and processes. As researchers continue to refine and expand upon this dataset, it is likely that our understanding of complex biological systems will continue to evolve, paving the way for groundbreaking discoveries and innovations in the life sciences.